All right. Good afternoon, everyone. Sorry to disturb your lunch here today. Yes. But I think we should get started. We have some great lineup of speakers, so hopefully they will have good time to talk about this. So good afternoon, everyone, and welcome. I'm Shashank Priya. I'm the VP for Research and Innovation, and it's my great pleasure to welcome you to this symposium. And if I pronounce it correctly, it's ProteXidine, right? Okay. Which is really, and the title is actually ProteXidine's, the intersection of science, technology, and skin aging. But I was just told there's like 5 other functions that this can do. So it's only very long title, so I think they just shrunk it, kept only 3 words there. But we're very, very delighted to host this event, and very grateful to see so many of our colleagues and partners and friends here in order to support the technology development and also celebrate this very remarkable scientific achievement. Actually, in my job, every day, almost every day, I get to hear some really great idea. This is probably the best part of my job, that in addition to solving termination orders and stop orders and all that stuff, but I get to meet some really talented people, and they tell me all the great stuff that they are doing. And you learn about so many different things that our faculty are exploring in all different disciplines, which will enable someday what this is doing now, impact the society and impact the people, and really help people who need some kind of assistance in order to solve the problem that they are having. And this is just the exciting part of it. Like once you see this, everything else makes sense, that I'm doing all that because this will be possible around it. So today's focus on this, and at this point I was told, don't call it drug, it's actually it's a luxury item. You need to have some money to buy this. But it's a very compelling example of how fundamental research, after some lot of persistent and hard work and a lot of sweat in the lab, can finally lead to some transformative solutions that can be utilized by general audience. ProteXidine is a patented biomimetic molecule developed at the Center for Drug Design, CDD, here at the University of Minnesota. This innovative compound enhances the skin's natural ability to manage UV-induced damage and reduce the effects of oxidative stress, advancing the science of skin aging and protection. The story of this medicine, or the cream, let's say, begins with the Center for Drug Design, founded in 2002 by Dr. Bob Vance, who continues to lead the center with high distinction. Under his leadership, CDD has produced over 35 US patents and discoveries, spanning anti-cancer, antiviral therapies to Alzheimer diagnostics, and now skincare science. Dr. Vince's past work led to the development of compounds foundational to, hopefully I'm pronouncing it correct, abacavir, a key anti-HIV medication marketed as Jizan, a discovery that has had global impact and brought significant revenues back to the university to reinvest in future innovation. ProteXidine continues that legacy. This molecule doesn't just treat symptoms, it supports the body's own regenerative mechanisms by helping removing damaged cells and signaling their replacement with healthy ones. We're very proud of this technology, not only about its scientific breakthrough and commercial success it will have, but also the science and innovation that went in developing this chemical composition. Also thanks to collaborative efforts of Dr. Abbas Raza, our technology commercialization team, and our partners at Topics Pharmaceuticals. This ProteXidine has been licensed and is moving towards commercialization and broader impact. Actually, this is the type of partnership that we are very proud of at the university. And I don't know if Rick is going to talk to you today, but Rick and his team, and I see Rebecca here, Ken, and Kevin, and so on, they have done just remarkable work. I came here from Penn State 2.5 years ago, and the first, when you do onboarding exercise, and they tell you that since 2006, we have launched 250 startups, and we are like the largest startup creator in the state of Minnesota. And now I think this number is 270 plus startups since 2006. And I think we are going to celebrate in few months 100 startups in last 5 companies. So if you're around, please join us in this big celebration. We'll have great food and drinks, I promise. But it's just amazing. Like, if you think about it, that this university has taken the funds from government, state and federal government, and that's our input. But the output is hundreds of parents each year, hundreds of licenses and deals each year, and 25 plus startups each year. So that impact is very visible that we make here. And I think it's all possible because we have quality people who utilize the resources and talent in a very, very effective way. And I think CDD is a very good example of this, that how continuously, year after year, and for decades now, they have provided breakthroughs. And not only leave it at the scientific level where you publish paper and you file patent, but actually find the industry partners, work together with them, solve the questions that they are asking, and ultimately transition it so that they can take it to that places. So this is a great story for University of Minnesota and good example of the culture that we have here. So I'm so proud of this, and thank you for inviting me to come here. And it's my great pleasure now to introduce our visionary scientist, Dr. Bob Vince. He's a distinguished medical chemist, inventor, and one of the most impactful researcher at our university. So please join me in welcoming Dr. Bob Vince.
Right here? Okay. Okay, thank you, Dr. Priya, for being here and also allowing us to present some of our technology to the rest of the university. I was asked to just give a few minutes on the history of our center and a little bit about what we're going to be talking about today. Now, I'll see if I can get my slides going here. Which one? This one. Didn't do anything. A little technical.
Technical glitch.
Difficult.
It's on PowerPoint, right?
You trying to get to the PowerPoint?
It is on. It's on the slideshow.
Yeah, let's go. Where's the, is it this one?
Yeah.
Is that it?
Yeah. This is,
That's it?
Does it work?
no.
Advance?
No.
Oh, let's try this one more time. And which one?
This, right. This is it.
No. He's trying to say.
Yeah.
I
Okay.
think we're good. Let's try restarting it.
Yeah, it's good. Yeah,
There you
it's
go.
moving. Either this or this.
Or press it. See what
Yeah.
happens.
Yeah.
Is that it?
Yeah.
Okay.
Just this one.
Okay. Yeah, it's hard for me to see here. Okay, so just this is our logo, the capsules, and okay, a little bit of the history. The Center for Drug Design was founded based on royalties that we received from a former invention, where the university received $600 million in royalties. And part of that came back to the inventor's program. And so it was about 25% of that. So we used that money to start our center and use the royalty money to hopefully develop more drugs and things that would bring in additional royalties and keep our center going. So the university doesn't pay for anything that we do. We pay all of our own salaries. and we get grants for all the research that we do. And when we started, we had about 60 people in our center. I think we have about 50 right now. Went down a little during COVID, and we're building back up again. So let's see what we have here. Actually, the center was suggested to me by the president of the university at the time, Dr. Mark Udall. He was worried that certain administrators were going to try to take this money away from us, and he suggested that we have this center, and we become part of the Academic Health Center, which was a big center at that time and included all of the health sciences, colleges, pharmacy, nursing, dentistry, et cetera. It was, and the senior vice president, Frank Serra, at the time, agreed with that, and so did the vice president for research, Christine Major, was here, and this was all approved by them. So we became part of the Academic Health Center at that point. And this is the drug that we developed. It was called SkinAging, and I think there were about 13 years left on the patent, so we didn't get the full patent life out of it, but during that 13 years, they sold over $8 billion worth of this drug, and our royalty share was $600 million. Okay. We had 8 research groups at the time, et cetera. I'm trying to speed through this a little. And this is something I'm proud of because when President Udall left here, the day that he left, he sent me an email, and I'm kind of proud of that. He says, I'm proud of some things all of us together have accomplished. Nothing brought more personal pride than your landmark achievements in the establishment of the Center for Drug Design. And he said, even as I sojourned down interstate, we'll keep in touch. And he did keep in touch. You know, I can picture him going down, pulling his furniture behind him with his car, going down interstate. Then he left here, he became president of the university in Texas, and then later the California system. And I think he's retired now. The SkinAging royalties, some of the things that were used, Chris Major, who was the vice president at the time, set up 21st Century Graduate Fellowship Endowment that was used throughout the university, McGuire Translational Research Facility. The College of Pharmacy, the dean's budget got $48 million. Department of Medicinal Chemistry, which I was in, we set up graduate fellowships. We used some of the money for infrastructure, and I asked that each faculty member get a $1,000 grant from this. And then Frank Serra set up the Robert Vince Chair Professorship in Medicinal Chemistry for $5 million. I never did take that. I actually gave that to Gunda Georg. So that's where her chair professorship came from. And we used $11 million to establish the Institute of Therapeutic Discovery and Development, which is part of the College of Pharmacy. And then the Office of Technology Commercialization, I don't know how much they used. They had about $200 million to work with. When I came here, there was only one person in that department. His name was Will Fornell, and he worked until he left here in the middle of about 1980s, and then they hired one person to replace him, and then a little later, 2 other people. So all the time that we were working on this project, there were only 3 people in the department, which we called OTC, Office of Technology Commercialization. So Kay Terry was the person who did most of the negotiations, and then there was a person named John Tinty and then Tony Strauss. And they were the three people in the department at that time. Okay. I want to talk about the two co-inventors here on ProteXidine. We're going to be talking about ProteXidine. Abbas Raza, Dr. Raza, and Christine Dries over there. We're all equal. There are three equal partners in this. And we came up with this idea, and we said, well, Abbas, if you could do the chemistry. And he did. He made these compounds. He had to keep adjusting the compounds so that they could be formulated into a cream, et cetera. Christine did a lot of work where she had to actually design new glassware and techniques to show that the compound actually protected DNA, repaired DNA. She worked with skin preparations, and so she has done a lot. So I just want to make sure that everybody knows who these, who the inventors are here. And then I want to thank the ICAL Office of Technology Commercialization. Rick Hipp, who's in charge of this group, and the people that have really helped us, not only on this, but throughout all of our inventions. Karen Oldfish, she does all of our patents, and then Kevin Anderson, who's over here, he's head of the associate director. Rebecca Gerber, she did worked with us even on this project a long time ago, and then she got promoted. But she did a lot of the original with licensing and that type of thing. And Ken Karinja, oh, here he is. He's the present person who's been working with Topics Company to for our license and the development of this. And Doug France, who is working with us on another project that we hope to have another one of these meetings later in the year, featuring our work on retinal scanning for the determination of early Alzheimer's disease. And that work is done mostly by Dr. Swati Moray, who's here. And there are about 10 clinical studies going on in that research right now. And we should be able to present, have a Retispec company come here later in the year and feature that technology. So anyway, that's all I have to say right now, and I'd like to have Dr. Raza come up here and introduce the speakers, okay?
Well, good morning, everyone. I can still say, because it's not afternoon yet, so good morning, everyone. And thanks, Bob, for the brief introduction of our center and our association with the technology commercialization. And that's really great. And we are really lucky to kind of work under that vision every day and try to motivate ourselves looking up to Bob. So that's our motivation there. So thanks, Dr. Priya, for making time to come over and just say a few words about the technology and our center. I appreciate it very much. Thank you. And I'm here to introduce our guests today, and they're distinguished experts in their area. And we have a talk that lined up back to back. So I don't want to kind of intrude in between and separate the speakers separately. Rather, I would just introduce them back to back. Then I would give the podium to the experts here. Well, thanks everyone for coming over and to listen to our story of ProteXidine here. So we are honored and pleased to have two distinguished experts with us today to share their insights on ProteXidine and its launch. And our first speaker is Steve Hernandez, the Senior Vice President of Topics Pharmaceuticals. And he's also the Senior Vice President of Research and Development at Topics Pharmaceuticals. He has been creating and formulating treatment products for dermatologists and consumer skincare for over 35 years. He leads a group of 30 chemists, biochemists, microbiologists, and support staff. He and his group has published regularly in peer-reviewed medical literature journals. His group's latest endeavor is unlocking the power of ProteXidine. And with that, I would like to transition to our next speaker, Dr. Naeem Isa. So we will hear from Dr. Naeem Isa, who is a board-certified dermatologist and the Director of Clinical Research and Academics with Forefront Dermatology and practicing in Vienna, Virginia. He has multiple affiliations. I think if you go down this list, it will take me forever to finish this, but I'll try to do my best. And he's also an assistant professor at the University of Miami, Dr. Philip Frost, Department of Dermatology and Cutaneous Surgery, and also a clinical assistant professor at the George Washington University School of Medicine and Health Sciences, and also a clinical instructor at the West Virginia School of Osteopathic Medicine. Dr. Isa has received his MD and PhD from Georgetown University School of Medicine in Washington, D.C. He completed his internship in general surgery at Washington Hospital Center. He then completed a postdoctoral research fellowship and dermatology residency at the University of Miami Jackson Memorial Hospital. His PhD is in tumor biology from within the Lombardi Comprehensive Cancer Center at Georgetown University Medical Center with a focus on computational methods for drug discovery. His work is particularly focused on repurposing current FDA-approved drugs for new dermatologic and cancer indications. Dr. Isa's research interests include drug discovery for hair loss, vascular malfunctions such as Sturge-Weber syndrome, and healing of chronic wounds. His major research accomplishments have been the repurposing of the anti-hookworm medication mebendazole as a kinase inhibitor and anti-angiogenic for various malignancies, and the discovery of the first small molecule drug to directly inhibit the JNAQ protein implicated in Sturge-Weber syndrome. Dr. Isa is a nationally and internationally recognized lecturer and has authored over 50 peer-reviewed publications and 2 textbooks chapters across various disciplines within dermatology and drug discovery. He has received research funding support from the Sturge-Weber Foundation, Dermatology Foundation, and the University of Miami Scientific Advisory Committee. He's also an inventor of a patent on CBD nanoparticles combined with minoxidil for hair regrowth, and a pending patent on a novel anesthetic combination for ambulatory skin surgeries designed to reduce the need for epinephrine. We are excited to have both of them here today, and please join me in welcoming Steve and Dr. Isa. And we have a Q&A session in the end, and if you would like to save your questions for the end, and we will have a mic that will go around, and it will be easy. I think Dr. Isa and Steve will be happy to take those questions for you at the end of the talk. With that, I would like to introduce Steve. All yours.
Okay. See if I can advance the slide. Very good. Thank you for your introduction, Dr. Raza. Thank you, Dr. Vince, and the team for the incredible technology that Kevin Anderson reminded us that we looked at. It's been a bit of a journey a couple of years ago, going back to 2018, in fact. And I really can't overstate the amazing impact that this technology has had in the dermatology world, even as we continue to roll it out. What separates this technology and makes it special, and I've seen a lot of different offerings, and I've worked in the industry for over 35 years in commercialization and adapting new molecules to for consumer use. What strikes me as a chemist is the absolute elegance of how the molecule was put together, how it was just stripped down to essence. Every piece of it is functional, and it does so many things, while so many competitive groups were having a lot of problems and really, just getting tied up in not seeing through and seeing the concept, and focusing on what was important. And it really strikes me as a chemist when the smoke clears to look at the molecule and look at what it does and say, well, that's vision that brought it there. So I'm very honored to speak before this group. A little bit about Topics Pharmaceuticals. Topics Pharmaceuticals is a company that's been around for about 40 years. What we do is we formulate for the dermatologist. That's our primary mission. We formulate drugs, office use products, medicines that the physician would only use in the office for primary treatment, as well as over-the-counter and also consumer products. We also have a division that contract manufactures. So when I said I've seen a lot of things, I've seen a lot of things. On the contract manufacturing side, we've produced thousands of formulas for other companies. We're vertically integrated completely, so we actually do the formulation for them. We'll work with concepts that they have, put together the formula, and then we'll do all the testing and all the required registrations, all that type of thing, safety testing, and then they would launch it. I was at the American Academy of Dermatology a couple of months ago, with Dr. Raza, as I walk around the convention floor, I see a lot of companies whose products we've actually formulated for them. So, and we do that quietly under non-disclosure agreements in the background. So Topics is out there more than you might think. Here are some of the real early discoveries that, or some of our early, very early wins at Topics, and I'll advance to the next slide. So glycolic acid, it became tremendously, tremendously popular going back to the original patents, Rui Van Yu and Eugene Van Scott, Rui Yu and Eugene Van Scott, and it really changed dermatology. The alpha hydroxy acids were multifunctional, and they did a lot of amazing things to the skin. The problem was that they were terribly irritating. And when we did our research into them, we realized real quickly that it wasn't the AHA itself that was irritating. It was the byproducts that came along when they were commercially produced. Things like formic acid, methoxyacetic acid, that couldn't be completely cleaned out of them. So we took this cosmetic concept up to drug standards, which is what Topics does, and provided a very clean molecule, a very clean source of it, formulated with that. And what was amazing was I got one of the best compliments of my career. An MD, PhD professor of medicine at SUNY Downstate asked me to make him a very high concentration product, and I did. And he came back to me and told me he didn't think it really had that concentration in it. So I said, well, why would you say that? And he says, because I don't see any redness, there's no irritation, there's no real evidence that it's there. I said, well, keep using it, you'll see the long-term evidence. Then I gave him an HPLC analytical method so that his graduate students can look at it and see, and lo and behold, he said, wow, it's amazing, it is there. So one of the fundamentals that Topics says is we bring cosmetics up to drug standards, and there's a very big impact from that. Next, L-ascorbic acid, real quickly, huge. When it went on Good Morning America back about 30 years ago, Duke University, they patent, Dr. Sheldon Pinnell and Douglas Starr patented the stabilization of L-ascorbic acid. You can look it up on uspto.gov. It's Starr and Pinnell 043. And when I got the patent and read it, the first thing I realized was that it deteriorated over 20% in 12 weeks in some of the data that they had. So I scratched my head and I realized, oh, stabilization to the patent office does not mean the same thing it does to the U.S. Food and Drug Administration. It has a very specific drug definition. So we sought out to make it so it would meet the drug definition, and Topics Pharmaceuticals has a patent on stabilization of L-ascorbic acid. We're in our products. It lasts for 2 years without turning color, and without deteriorating. Next, antioxidants. We took a look real quickly at what was going on. A lot of buzz in antioxidants and how amazing they were, but we noticed that in the cosmetics world, they were using green tea extract. The extract has very little of the active ingredient that you actually see in the medical literature. So what we did was we actually worked with the concentrated polyphenol isolate at the levels that you saw in the medical literature, and we were able to get amazing results in terms of redness, anti-irritancy, and all the benefits that were published. And there's a long path to get there. They're very hard to stabilize, especially in water-based products. Next, we did a lot of work with delivery systems, and I'm proud to say a couple of years back, we were able to publish that we took retinol, which is just a vitamin A, it's what you would take in a vitamin supplement, and by using a proper drug delivery system, we were able to get it to induce the same effects on the skin that retinoic acid will. Retinoic acid is retin A, or generically tretinoin, which is a prescription drug. So by having the right delivery system and understanding the chemistry, you can get the non-prescription, in some cases, in the retinol case, to work like the prescription drug. So delivery systems was something that very much overlooked in cosmetics. And again, Topics likes to bring things up to drug standards. So moving forward, this is kind of our focus, what we like to do. So what we do is we're really efficacy driven. I produce a lot of products that go into consumer markets, and in the consumer markets, they are trend driven. So everybody's looking for what's the next big thing. Is it the snow mushroom, or is it ashwagandha, or this or that? You know, they're looking for the next herb, the next buzzword. In dermatology, it's all efficacy. The doctors want to cure the problem. So that's why we focus on really delivery systems and active ingredients and getting things together that'll get us there. We love to publish in peer-reviewed medical literature, and we have a network of over 3,000 dermatologists. So when we give things out, we get feedback from the field, and we're able to fine-tune it and work with our derm network to actually optimize the products. Here are some of the examples of what we've published in. So we've published in with regard to using antioxidants, Dr. Neil Brody there, using the green tea polyphenols and resveratrol, but not in the ways that you'd see in the consumer markets, in very high concentrations like you see in medical literature, and it was very useful against rosacea and inflammatory conditions. Dr. Michael Gold, we work with peptides, and he was able to do a study where he did RF microneedling. That's where they shove needles into the skin and then turn on an RF current between the needles that kind of cooks the skin. And what it does is it makes the skin believe that it's just been totally ripped off, that it is destroyed. And what it does, and it tricks the skin into believing that, which sends a whole cascade of chemical messages to make new collagen and repair itself, and the effect is quite good. But the problem is the patients are miserable when they go through that. And using our techniques and our products, we're able to shut down the irritation, shut down the erythema, stop the swelling, and really made great strides without using medications that sometimes would work against the patient or would be contraindicated. Dr. Goldman did a big study for us on use of signaling peptides in order to improve the appearance of the face and neck. Dr. Dralos is the one who did that retinoic acid study for us. Dr. Jadego, he had published a vitamin C study for us, and Dr. Issa, who's here today. Just amazing work, and the latest publication is in Journal of Drugs and Dermatology on ProteXidine, and it talks about how the many features of the molecule and how well it works. One of the things that was interesting is some of the other groups were claiming that they can prevent CPDs using photolyases and things like that. We would assay the products, and I couldn't really find photolyase in there, but my assay methods were not really as delicate and as able to detect in the micro quantities as they could be. I spoke to many commercial laboratories out there to see if they could get an assay. SGS, Eurofins, the big names. Nobody could come up with anything plausible. Talked to Dr. Raza, and 3 hours later, he's on the phone with me, mapping out the most amazing assay. And I'm following it along, and I'm going, unbelievable. I spent 3 weeks on the phone with the major commercial companies, couldn't do this, and he just sat down and boom. So it just shows you that when you have command over the material and the chemistry, you can do amazing things. So one last thing that's really interesting about us at Topics is we're fully integrated vertically. So that means we have an analytical chemistry lab, we have a microbiology lab that's headed up by a PhD microbiologist. So as we go down the line, there's a laminar flow hood there. We do all our own microbiology work. Next that's a high shear Silverson mixer. Interesting. You can give somebody something and tell them it's good for them, but you won't get compliance unless it feels really nice and unless they see something right away. So the aesthetics are super important. So we actually work on making the medicinals as well as the cosmetics have a lot of cosmetic appeal because that brings compliance. When you get compliance, you get the result from the medication. That's the picture next to it is high performance liquid chromatography. We do a lot of work on stability studies. That will give you a nice signal showing you if everything's still intact or if it's breaking down. We also use it to do a little competitor intelligence. So we'll go in the field sometimes and collect samples and see what they're putting in their products. At the end is one of my favorite pieces of machinery. It's been tremendously helpful in dermatology, and that's a DNA analyzer. And what we're able to do there is take a formulation and look into it and see, instead of just putting a textbook amount of preservatives, and textbook preservatives are really made for shelf life. So they're kind of, I call it embalming. You put so much preservative in there that it's going to have a real long shelf life. And you don't worry about the 2% of the population who might have a reaction to it. Well, since we sell mostly to dermatologists, that 2% turns into 20 or 30% because these people at the derm office has just been lasered, microdermabrasion, maybe one of Dr. Gold's patients that just got some RF microneedling. So they are super sensitive for months after that. And so what we're able to do is check the formulations we make, see what actually is able to grow in them in terms of microbes, and then strategically target only those microbes. Sometimes we can come out successfully passing all the government testing with 10 times less preservative than you would find in a standard textbook. And that's tremendously helpful in dermatology and some other special markets like infants. Infants have a different surface area to body mass ratio to their skin, and they can also become very sensitized very easily. So, and also in the geriatric market, the elderly don't clear medications from the skin, and they tend to build up. So, as well as preservatives. So we're able to use, all of these to really focus on formulating specifically for the derm community. And then when you take those products out of the dermatology community, they've already been optimized for the general public. And we've seen that when we do a lot of work over in Asia, China, Japan, Korea, very sensitive skin populations, and they don't have problems with our product because they've already been optimized for sensitive skin. So just a pleasure to be working with ProteXidine. The early feedback is amazing, and one of the early challenges we had with some of the internal people were concerned about the chemistry. It's a long explanation, cyclopyrimidine dimers and all that stuff. And I remember the first time I presented it to Dr. Issa, I was about 3 sentences into my explanation over the biopsy when he just said, "Stop, stop. Let me ask you a question. Did your product do that?" And I was like, "Yes, it did." And he instantly said, "Okay, I want to work with you guys. I have to understand more of how this works." And he became our speaker, and he's an expert in cancer and photobiology. So it was a great fit. But that took away the question real quickly. How do we explain to people this complicated technology? Well, our customer base is physicians. And when we got that reaction was reported back. We knew we had to get behind ProteXidine in an even bigger way. And as we move forward, we just got approval to sell in China, and the US and everything has been really moving well. So I'm just super thankful. This is the type of product that has two functions. One is as a cosmetic, and then the other, and you'll see some wrinkle reduction pictures and some interesting collagen stimulation pictures. So we have these cosmetic effects, but also Dr. Issa will be presenting forward-looking data, meaning ongoing research into future drug applications or possible drug applications of the amazing technology. So it's both has cosmetic application as well as future drug application. And with that, I'll turn it over to Dr. Isa.
Well, thanks for having me, and great, Steve. You know, Steve is a visionary. For those of you who haven't had a chance to meet him, like, he really was at the helm of topics, you know, developing, you know, the R&D and the vision for all of this. So if you get a chance to talk to him, he's an excellent person to meet. So again, my name is Naeem. It's a real pleasure to be here and to speak to all of you in the middle of your day on a Thursday, right? I made it this far, so before the carbs kick in and the sleepiness, right? I'll just tell you that there's one theme, you know, as you've seen here, especially at the University of Minnesota, you know, with Dr. Bob Vince, you know, with Dr. Raza, Dr. Swati, so on and so forth, is that here you are, I would say, in a mecca of like academia and vision and science. Each one of you in this room right now is working on something potentially remarkable, all right? And really truly believe in your science. And in order to get past to the next level in whatever you're working on, you have to sell your science. You got to understand your science in the complex way, but also for dummies, all right? And so the thing is, if you're able to explain your science in literally 60 seconds or less and why it's important, you win the game. So that's just one nugget from my personal experience in struggling for nearly 2 decades of trying to explain. You know, I did a lot of computational work, you know, drug repurposing, trying to say I have an algorithm to give you this, that, and the other thing. And people would say, who cares, right? So who cares about your science, right? Everyone around you here does care, but how does the outside world care, right? And how can you translate that for something that patients will care about, so the academic institutions will care about, you know, and ultimately for yourselves and humanity, all right? So just keep that in mind, you know, as we move on to ProteXidine, because ProteXidine is really quite a remarkable feat for something that may seem complex, but once you see how simple it is and how you can communicate that, you know, the world's your oyster, right? So on that note, I'm happy to be here to talk to you about ProteXidine. Here are my disclosures. I talk with a number of pharmaceutical companies and am on their advisory boards, including topics. But let's start with the basics, right? So, has anyone here experienced sunlight? Right, you're humans, you know, you live on planet Earth, right? This is how we live, breathe, you know, plants photosynthesize, all that stuff. Sun, while it gives life, can also be dangerous, all right? And from the dermatologic perspective, the sun is not your friend most of the time. Other times it's great, but most of the time it's not your friend. So if you look at the electromagnetic spectrum, thinking back to physics 101, right? You can break down the electromagnetic spectrum to a couple of major sections, but 3 which we really care about in the dermatologic world. You have the ultraviolet spectrum, right? You have UVC, which is blocked by the ozone layer. You have ultraviolet B, which could cause skin cancer, ultraviolet A as well. Then you have your visible spectrum and infrared spectrum. Focusing on ultraviolet, you get a couple of different skin conditions. So actinic damage means sun damage, and that gives rise to the precancers. For those of you who have had a lot of sun damage throughout the years, putting on that banana boat, tanning oil, right? Putting on that iodine, sitting and baking. If you rub your hands across your forehead, you feel some gritty sensations. Those are the precancers. That's from ultraviolet light, right? And if you feel that, go to the dermatologist. Number 1. Number 2, you can also have skin cancers as well as the photodermatoses. So photodermatoses means that you have these autoimmune kind of components. So lupus can be exacerbated from that, or polymorphous light eruption, so on and so forth. Then you move to the visible spectrum, right? So this is interesting. So the Roy G. Biv, right? This is your rainbow. This is the visible light that, the colors. If you don't protect from that, actually, you can get discoloration as well as skin disease. And a lot of the photoprotective options that we have out here are just mostly focused on ultraviolet protection. They say broad spectrum sunscreen or SPF or what have you. That's only a part of the spectrum. So you have the visible light that's still doing damage. So we need to protect with tint, for example. So you have iron oxide and what have you. That helps with these other discolorations and other skin conditions. And for infrared light, we're still figuring out what that does in the skin. And just a quick note on blue light, there's always these TikTok trends, right? You see on YouTube or whatever, Instagram, that, you have to protect from blue light. Guess what? There's no evidence for blue light actually, causing damage to date. So how does ultraviolet light specifically, cause damage? You have different wavelengths of light, and without getting too nitty-gritty into the physics part, is that different wavelengths penetrate to different aspects of the skin. Ultraviolet light can go deeper in the skin, ultraviolet B can go a little bit more superficial. But what happens is that these are both damaging, and if you go deeper, you're affecting skin collagen and elastin. This is your framework, right, of the structure of the skin. That gives you beauty, gives you tautness, right? But then you also have a little bit further up, when you get to the epidermal layer, you have some melanocytes that give you color. That's where you get your pigmentation changes or pigmentary disorders, darkness, lightness, and what have you. You also have oxygen in there, so you can generate reactive oxygen species. These are the electrons hanging out on the oxygen that go and damage your organelles, lipid membranes, so on and so forth. And that causes a cascade of reactions. And then you can also have generation of what's called the CPDs. These are your nucleotide dimers that cause errors and mutations as part of repair, leading to skin cancers. Here's an example of ultraviolet light getting in and causing a chain reaction with your reactive oxygen species. You create one ROS, you make others from that, you have a cascading effect, so on and so forth. No, you see it says SPF at the top there, that an SPF or sun protective factor can absorb approximately 92 to 98%. So have you guys seen the word SPF? All right. Just a shout out from the folks in the room here. How much SPF is recommended by a dermatologist, do you think? Any number that you may see on the bottles. I'm hearing 15 and 30s. Definitely not 15, all right? You want to go 30 and up. 30 puts you in that 92% range, okay? But SPF is only measuring ultraviolet B. It says nothing about ultraviolet A. So if you're focusing on sunscreen that does not say broad spectrum, you're focusing on UVB. And as we just said, if I go one slide before, UVA, right, it goes everywhere, all right? It does a lot of other damage that you're not protecting against. Furthermore, even if you have a broad spectrum sunscreen, you see that you're not getting 100% coverage, all right? So by having less than 100% coverage, these rays will go and cause reactive oxygen species, the dimers, and so on and so forth, all right? So you may think that you're protecting, 100%, even if you're slathering every 30 minutes, all right? But we're still not, because we're not getting 100% coverage all the way. And this is an example of a bad sunburn, all right? Now, it's pretty terrible. It hurts, it stings, and the statistics go that one bad blistering sunburn can increase your risk of the most common skin cancer called a basal cell carcinoma by 1,000-fold. So for all of you who either have teenagers or were a teenager who had a bad blistering sunburn, you're at risk 1,000-fold. It's almost like being in a tanning bed for melanoma, okay? This is, so again, public service announcement, right? And so even if you protect it, remember, we talked about SPF, right? You can spend more time in the sun because of the SPF. You can be at the beach and what have you. But you don't necessarily have to feel the burn in order to get the damage. That's the point. So understanding that understands the need for other photoprotective measures and repair measures, and hopefully having that as an over-the-counter rather than a need for a prescription and going through the FDA. And that's exactly what ProteXidine does, which we'll get into the science momentarily. This is my favorite image that I show patients. This was not only featured in the New England Journal of Medicine, but this was also shown in Time magazine, I think about like 5 years ago or so. This is a patient, Caucasian, trucker, who was on the road for 28 years, did not protect the left side of the face, all right? Clearly, this is not fake, this is not Photoshop. This individual literally aged the left side of their face because the window does not protect you from ultraviolet rays, right, and so on and so forth. So now, go ahead and look at yourselves in the mirror, when you go home. See if you have more sun damage on the left side of your face or your arm where you hang it out, from the window and so on and so forth. Again, very real damage. And some, cutaneous manifestation of this damage is you can get wrinkling, earlier wrinkling as well. You can have changes in the blood vessels, so you can get redness and ruddiness, and they call it poikiloderma for those who want to be fancy. You can have that melasma, the dyspigmentation in that middle section there. You can see more melasma on different skin types, and again, more perioral rhytids or grooves and what have you, collagen disorders, etc. So, for all you who are scientists in this room, all right, so it sounds great, how do you make sense of how do you want to protect against sun damage? My background, I was a computational chemist or pharmacologist. Things had to fit in boxes for me. Medicine has to fit in a box for me. No pun intended, like to get it in a box, right? The way to think, right? So I was a programmer, things had to, fit in an algorithm. First, we talked about protecting ultraviolet A and ultraviolet B. So you have your filters, and there's all sorts of filters out there. We're talking about we're talking about zinc oxide, titanium oxide. You can also have those in Europe that can also use like fern extracts and natural supplements, so on and so forth. Then we talked about the visible light, right? And this is where you have your tint and your iron oxide to block that color spectrum to help with pigmentation. Great, you can put these in there together, fine. Then, remember the reactive oxygen species. You want to protect against that cascade of oxygen and the electron damage. Great, you can add some antioxidants there. Then, let's say that you go through all of that and then you still have damage, right? And you're making this DNA damage. So you want to repair that. So you can add DNA repair enzymes to that. And there are some products out there that contain DNA repair enzymes, but they have some challenges as well because they're enzymes. Like good luck manufacturing a well-folded protein in the right states with good manufacturing protocol and then shipping it, putting it on the shelf, and getting it into people's hands, right? That is the supply chain problem there. And furthermore, you can think about add-ons. You can have anti-aging ingredients such as a retinol. You can add some exosomes, which is the buzzword, but it's not regulated at all, so be careful, with what you're getting. And then you have growth factors, hyaluronic acid, so on and so forth. So you can cook with this, all right? There's different ways of cooking with this. Talking about the reactive oxygen species and antioxidants, right? So you have some naturally occurring antioxidants, in your skin, in your body. You have enzymes such as superoxide dismutase that takes superoxide. Then you also have catalase, which takes away the hydrogen peroxide that is damaging to membranes. And so when you have ultraviolet radiation, you can deplete these enzymes. That's what's called oxidative stress, and leads to all these, conditions that you all are studying. How can you counteract that specific part? Well, you can increase the expression of those enzymes like superoxide dismutase as well as catalase. Some antioxidants in of themselves, such as coenzyme 10, all right, CoQ10, which is affecting the respiratory chain of the mitochondria, that can also increase the expression as well, for those who didn't know that. You can also increase proteins such as heat shock protein that can protect the proteins and the folding in this kind of stress environment. You can also apply, as we said, the exogenous enzymes. You can produce it in a lab or some manufacturer and put it on there. But they're difficult to formulate, and you may have regulatory hurdles. Especially depends on what source that you're using. If you're using a mammalian source, if you're using a bacterial source, or even a human source. You can also develop mimetics from bacteria as well, such as the Thermus thermophile bacteria. Again, it's a difficult thing to accomplish, right? So that's one aspect, right? The reactive oxygen species. Then the other part is the actual DNA damage. And you have another set of enzymes called lyases. And specifically, there's one type called a photolyase. Photolyase is an enzyme that will go and repair the damage. It has to be activated by light, which is also ironic because you want to protect yourself from the light, right? So getting that is also difficult if you want to put a photolyase in. And humans actually don't make photolyases. Humans, in order to get a photolyase, you have to get it from a different eukaryotic organism and manufacture and put it on there too. But you will see that some products may contain that in their sunscreen and other topical products. Then ultimately, you can consider recruiting the enzymes that are going to help repair the actual damage. So there's a whole section called the nucleotide excision repair, which we'll get into momentarily. Here's some examples of DNA repair enzymes. We talked about photolyase. You also have UV endonuclease. You have OGG1, T4 endonuclease 5. These are some of the brand names that are, or trademark names, I should say, as well as their sources, such as plankton extract, micrococcus, and so on and so forth. This is all there is on the market right now. It's a very limited space, right? And then you can't actually, it's hard to find out, as Steve was saying, how much is actually in the product. There is no regulation, so you don't even know what you're getting. So the major unmet need right now in photoprotection is novel molecules that can reliably promote antioxidation as well as DNA repair, and to do this in the presence as well as the absence of UV light. Because you want to put something on at nighttime, for example, and still get the same type of effect. Just a tribute, to my overlords. All right, these individuals are brilliant science. Dr. Abbas Raza, Dr. Robert Vince, right, and Christine Dries. These inventors really put in the sweat equity into this. They've really developed this technology, and I can't do as much service, to this remarkable invention. So please, thank them for their work, because this is truly a milestone, not just in dermatology and in cosmeceutical space, but in medicine, period. Especially, a tribute to Dr. Bob Vince, right? So he came up with the Vince lactam, right? And so this critical scaffold has led to the discovery of a number of, pharmaceutical agents that we use, at least in my practice and for those of you who practice medicine out there, on a day-to-day basis, right? And one of them is abacavir, right? One of the first HIV medications that has saved millions and millions of lives. This originated right here. Thank you, Dr. Vance. This is really a remarkable thing, just to think about that history. And furthermore, on a separate note of taking nucleosides and creating a structure called acyclonucleosides, which helps with production and manufacturing and so on and so forth. And so by creating an adenosine acyclonucleoside, that has led to the development of other ones that we use, such as the antiherpetic medications such as acyclovir and so on and so forth. So getting back to UV radiation damage, right? UV radiation, we're ad nauseum, right? I'm telling you over and over again, it's gonna get into your skin no matter what you're doing. You're doing as best as you can, but still getting in the skin. You're gonna have the reactive oxygen species, and you're also developing the cyclobutane pyrimidine dimers, or CPDs. These dimers is what causes the mutations, all right? So there was early work in the 1990s where someone thought about using thiamine dimers. So specifically, if you're looking, if you're studying your biochemistry and biology, you think about thiamine dimers specifically. When you have the UV damage, you make the dimers, and then you have errors in replication. When you have a thiamine dimer, that can actually cause the initiation of your nucleotide excision repair. So an individual back in the 90s said, okay, what if we just took a thiamine dimer, all right, and by itself, and stuck it onto cells? Can you actually recapitulate an endogenous recruitment of DNA repair? And that actually was proven true in the late 1990s. Taking just thiamine dimer, putting it on cells without light caused the endogenous repair to be able to come to the fore. However, what sounds great, and it's a small molecule and what have you, it is unstable, it is very expensive, and is difficult to manufacture. So they tried for over 20 years to get simple thiamine dimers into the market, but failed. It was a logistical nightmare it could not get overcome. So this is just an example of what a thiamine dimer may look like as a cartoon. I'm sure that's what it looks like inside your cell, but even more realistic here. So you have two thiamines that are next to each other, right? So you have ultraviolet damage, right? And then once you have the ultraviolet damage, they dimerize, and then you have the repair enzyme comes. The repair enzyme comes and cuts, removes, and then you have a ligase, put it together. Again, biology 101. You want to get those enzymes to rock and roll, right? So with that in mind, all right, recruiting the endogenous enzyme from the data that we have from the thiamine dimers, could there be a better way of making a stable type of thiamine dimer as a new molecular entity? And in fact, the team here, right, they have been able to do that, all right? So about maybe 9 to 10 years ago, actually, this idea originated. And so what they did was they made it acycloform. So remember, we were talking about, acyclovir in a few slides prior, and taking an acyclonucleoside. That same concept was replicated here for the thiamines. And they were able to make a low-cost, easily manufactured, replicable, and sustainable method to make this acyclic thiamine type of dimer. And by doing that, the thiamine actually, so let me take a step back, it actually doesn't, it is attached to each other, but will dimerize with an extra bond in the presence of sun, all right? So why is that even important, all right? The idea is that you want something to protect your own DNA. So this structure, if we go back here, this ProteXidine structure, acyclothymidine dinucleoside, you're never going to remember that latter part in there, so call it ProteXidine, all right? So ProteXidine, without ProteXidine, you will have UV damage leading to a thiamine dimer, right? So this is a schematic of that experiment that actually Christine helped to develop. But when you put ProteXidine, it takes that sunlight and dimerizes and makes that extra bond itself, kind of like a protectant, for your own DNA. But not only that, it will go and actually activate your repair enzymes. So this is a nice schematic, but did it actually do that? Well, yes. I hate looking at blots, all right? I thought I put that way behind me, all right? But it comes back to haunt me. But again, very elegant type of experiment here, looking at the chambers that the team here had created in order to make this experiment happen. at the top, you see the gel that shows you have your DNA that was put into the chamber. They shined light on it, ultraviolet light, and you see that there are different fragments. So if I move the mouse, at 0 minutes, there was just one fragment, one DNA molecule. And then about 10 minutes later, your nucleotide excision repair was already rocking and rolling, right? So you have some fragment of the original, and then you have other fragments of the cut up DNA, right? That's trying to get repaired. So you'll see more than one band. Then you add ProteXidine, and you repeat the experiment again. And here what you see is that you see the same band at 0 minutes, 10 minutes, so on and so forth. That means that you have protected against making the thymine dimers, and no nucleotide excision repair was needed, actually, for that specific experiment. So that was the first proof of concept there. The second, so that was just, in vitro, right? But who cares about in vitro, right? We want skin, give us skin, right? So here we actually have human abdominal skin explants. So where do we get skin from to do all these experiments, right? It's tummy tucks, right? And so literally the plastic surgeons down the road, what are you gonna do with all of that extra meat, right? You give to the university, right? So that they can study it. So what happens is you do the same kind of experiment. So on the left here, you're going to shine the light lamp, right? It has ultraviolet light in it. And the red immunohistochemistry is showing these dimers, right? These CPDs. It's a lot of them. You shine it, a couple of minutes later, 30 minutes, or what have you, you start to see all of these dimers. But if you have ProteXidine-treated human abdominal explant skin, you see that the amount that is actually produced is much less. So this is an in vivo, very realistic proof of concept showing that you're protecting against the ultraviolet light. This is more some electron microscopy here that you're seeing as soon as about 1 hour in after shining light on this abdominal explant with no ProteXidine, you start to see the CPDs, and then 8 hours later, it's CPD city. It's all white everywhere, all right? And then 24 hours later, you're seeing some of that clearance because your DNA nucleotide excision repair is kicking in. But what about when you're adding ProteXidine? At 1 hour, a very minimal CPD production. At 8 hours, minimal CPD production, and then at 24 hours, it's all cleared, right? So you can see it's a night and day difference between the ProteXidine treated and the vehicle treated abdominal human explanted skin. And actually, this is, for me, is the most exciting part here. It's like, yes, you showed that you can protect against the damage. You're showing that you could remove you could get the repair. But what is that in between, right? You want to, as part of your theses, right, or whatever that you're working on, you want to show that like true mechanism, right? The mechanism here is that truly you are recruiting the endogenous DNA repair enzyme. And actually, this is not even published yet. This is confidential information that we're showing up here, is that you're getting a higher expression of this enzyme here. And for those who are astute in the room, right, which is every single one of you, you probably have heard of different conditions or can imagine conditions where you may have issues with repair enzymes leading to different disease manifestations. And we can theorize that we may harness this technology furthermore to look into those kind of conditions and maybe helping to treat those, right? So maybe more to come in the future, from this kind of tech. Great, so we have the proof of concept, we did stuff in the lab, all right? So where does it go next? Then you formulate it, right? So you have ProteXidine, then Topics comes in, Steve and Abbas meet, and they said, okay, let's make some cool stuff before we talk to their dermatologist. So they made a couple of different formulations, some with sunscreen, some with like anti-aging properties, and so on and so forth. Then they showed me the data and said, okay, we did the abdominal skin explants with the 4 different formulations. Can you tell us what the data looks like? Well, again, in every single formulation that is dermatology relevant for what I can put on your skin, we were able to recapitulate that those skins that were treated with ProteXidine had less CPD formation and they were clearing after 12 hours. You see a theme here? Less CPD, more clearance with the ProteXidine. It's as simple as that, but powerful. Simple, but powerful. But if you do have some damaged cells, right? Let's say, okay, look, nothing is perfect, right? What happens to damaged cells or apoptotic cells, right? Because what happens is that when you have DNA damage, right, the cell's either going to turn into cancer or they're going to want to try to clear. Well, with the ProteXidine-treated skin, you see that those apoptotic cells are getting cleared real fast, right? Within 12 hours, you see those pycnotic nuclei getting right out of there. And that's exactly what you want, because you don't want necrosis. You don't want a fiery death of these these irradiated keratinocytes. You want them nice, clean, and get them out of there. And you really see that with the ProteXidine-treated skin. So that will briefly shift gears. So we talked about like, mutational damage, DNA repair, and what have you. But what about just like your collagen, right? We talked about that depth of the skin. We have the collagen and elastin in your skin structure, right? So but as part of aging, right? Aging can happen in two ways. You have your intrinsic aging. This is just the way you're built, right? At some point, we're just going to age, even if you sat within four walls for your entire life. It's your biological clock, right? But also if you have extrinsic exposures, these such as pollution and UV light and so on and so forth, and stress, etc. Glycation from eating sugars, like the meal that we all just ate, is going to make you age, right? I'm going to eat it too, don't worry. But so we're all going to age from this stuff. So can we combat these changes specifically even deeper in the skin with collagen and elastin? So what does that look when you have ultraviolet damage onto skin? All right, this is, again, human abdominal explanted skin. This is normal skin on the left here, showing what's called a basket weave pattern. Things are nice and tidy and layered on top of each other. Gorgeous. Then you show them some light. Everything is disarrayed. Your collagen is not really nicely layered. Everything's in disarray. You can see some weird elastin on the bottom here in yellow, and then you can see the keratinocytes kind of acting up in the red at the top there. But if you give ProteXidine and you shine the light as well, you're going to have the recapitulation of that basket weave pattern. You still have that collagen and elastin structure. So this is showing you a different part of what the molecule does. So not only the reactive oxygen species and the DNA damage and what have you, you also are helping with the collagen and the structure from below. So it's a 1-2 punch. We actually did a podcast on these results in the Journal of Drug and Dermatology. And who doesn't love a good podcast, right? We actually, when we published this paper, first of all, nobody reads anymore, right? Like you have like a million journals. Which one are you going to read, so on and so forth? Especially like with my colleagues in the dermatology space. You want something to really hit and gather attention. When we did this podcast, right? It's a 5-minute podcast, and anybody can listen to it. The predicted measure, for those of you who do social media at all, was about 400 views for the first 30 days, and for the entire year was expected 800 views. Within the first month, we got over 1,100 views, over 175% as expected. The need for something like this is out there. Every single person is interested. I'm sure you are interested for yourselves or for a loved one. And this is why this technology is important. And not only is the technology is important, it's also safe, right? Won't bog you down with different safety profile assays, but the topic specifically has helped to do a lot of these safety profile tests, so on and so forth. And you can see there's a number of tests. It went through the battery before we got out there. Something that is necessary, but they pass all of the marks, and that's why it's on the market right now. So how does it look, all right? So we talk the talk, does it walk the walk? And indeed it does. Here you have a formulation called ProteXidine Defense, and ProteXidine also can play nice in the sandbox because you can also include a number of other ingredients such as arbutin and maybe retinol and what have you, and it can be stabilized together to give you the ultimate products. So here you can see the periocular rhytids or wrinkles around the eyes, and so you can see that at baseline and what happens after that. All right, this is 12-week data. Imagine, you didn't do any Botox, right? You didn't do microneedling, you didn't do any fractional laser. You gave a topical, right? Consistent topical in the morning and in the evening, and you're able to reverse that damage. I have never seen such a product in my career. These are some side-by-side pictures, right? 12-week data. 3 months is nothing. It's a quarter of the year, right? And it's cheaper than Botox. What about on the nose? You can see damage and telangiectasias on the nose in this patient. Baseline, 12 weeks later. Again, not Photoshop. Obviously, you can Photoshop everything, but not allowed. So here you can see a side-by-side picture. Skin even looks less red than before, and smoother and so on and so forth. I really like this one, actually. It's probably my favorite one here. So this is a patient who has we consider skin of color, especially so Asian patients, while they can have Caucasian-looking skin, they're actually called a skin type 3, and so they are considered skin of color. And you can see the melasma, at the top and the rhytids and so on and so forth. And you can see over time, sorry, it didn't include the weeks, but it's week 0, week 4, and week 12. And you can see over time, on the left side here, this is the baseline. You can even see like these, these marks right here. This hyperpigmentary marks and melasma start to melt away, and you get smoother skin. So, according to the clinical trials, 100% of patients showed improvement in fine lines and wrinkles. 90% showed improvement in hyperpigmentation, and 87% showed improvement in skin tone. All of the major markers when you're looking into the cosmeceutical space. So what does the data currently suggest for ProteXidine? It prevents most of the dimers from forming. It also repairs and clears the CPDs that do form faster than if you didn't have it. It also prevents damaged cells, as we've shown, and also clears them, right? Those pycnotic cells, get them out quickly so they don't die a fiery death. And also maintains your collagen structure and your density and helps to build that up. Here's another formulation, right? So that one was called ProteXidine Defense, and I believe that's the one that's in your little packets there, so you can go ahead and try. Now here you have an age-defying eye cream. I actually gave this to my mother. It's not my mother on the screen, but I gave it to my mother, right? And I said, try that. You know, she's a little worn down maybe from me. But the thing is that her eyes started to look like these results in just 4 weeks, right? You can see on this microscopy here on the left side, look, it looks like the Sahara Desert, right? You have all the cracks here, you know, it's not, it's needing hydration and so on. But 1 month later, like we don't typically look at endpoints in dermatology or in clinical trials at 4 weeks. We look at 12 weeks, 16 weeks, 52 weeks. 4-week data for iritis or wrinkles, phenomenal. This is the age-defying cream, all right? It's out there, get your hands on it. If you don't have a dermatologist, get one and ask them for it. This is the discoloration defense. This is what it looks like. This is the one that's in your packets. And some of the advertising materials by our friends at Topics. And don't forget, a tribute to Topics is that they formulate and they do science really, really well, you know, with these assets. So they took ProteXidine and they gave you, you know, the discoloration defense and the age-defying cream, but they also have these other, you know, the vitamin C, you know, that's been stabilized for 2 years, which is remarkable, the glycolic acid. They have a whole array of products, you know, at your disposal. And then this is how the cream works for discoloration defense, for protecting the defense. Again, very elegant. He showed you that device, you know, for that formulation. If it's an ugly thing, you're not going to put it on your skin, right? Let's be real. Like, you're not going to put something that doesn't feel nice, is not elegant. This feels really nice, especially for someone like me. Look, I'm hairy. All right, I put this thing, that's not on my face, I put it on my hands to protect, and on my beard, and so on and so forth. And it works, it applies just great. And on that note, ladies and gentlemen, thank you for listening to my ramblings, and I am happy to take any questions that you may have. And thank you all for inviting me to this talk. All right? Wonderful.
If you have any questions, just stand up and raise your hand.
Hello, thank you for the wonderful talk. I just have one question about your in vitro studies. Have you performed France diffusion assay as part of your preclinical study to test the in vitro permeability and release of active form formulation?
Yes, we have. And as a matter of fact, a lot of that work was done here at the university.
Okay.
So yes, we have, and a lot of that work was done here at the university. And that was one of the things that originally attracted us to license the molecule and actually try to, you know, work with it and commercialize it, was that the university had done, I mean, it was a complete package with a bow on it. They had invented the molecule, had done some safety studies, some proof of concept. So when we came out and looked at it, we said, we can work with this. So good question.
Okay, thank you.
Does this product work on age spots? I got some here if you want to try it.
If you're talking about the stuck-on ones, that's a different one. But if you have like things that are just pigmentary, yes. Happy to look at it later.
Yeah, one thing I should mention there is that it was also formulated to be used with very popular dermatological regimens. So, for example, this one that's called ProteXidine Defense was originally called Discoloration Defense, and it was part of a melasma or a skin lightening regimen. And because of its properties and the way that it works cosmetically with UV light. So it's used in a lot of different types of regimens, and it's multifunctional, and that's one of the things we really like about it.
I was just going to say thanks so much for your talk, and I'm excited, your talks. I'm really excited to try my sample. But I did want to ask, this is kind of unrelated, but I couldn't help but notice up there the ingredient list, the hot ingredients, the exosomes, and the Arabidopsis thaliana. And I did my PhD in Arabidopsis, so I had to giggle, because I actually saw that on a product about 6 months ago, and I was like, what is that? Do you know anything about that? They're just grinding up plants and...
So sometimes they have these extracts, right? That they say that magically, if you dump the extract on there, that you may have certain, changes in appearance, right? And a lot of it is hocus pocus.
But it's completely unregulated. I mean,
completely, because it's in this case, it's in the cosmeceutical space, right?
Okay.
If they start to say that you have X, Y, and Z molecule that's going to alter the structure and function of some component of your cells, you put yourself in the pharmaceutical drug world, all right? And then that has a whole nother slew of regulatory hurdles and questions as well. So it's an interesting question that begs a lot of research.
Yeah, how do consumers protect themselves? I mean, obviously there's a lot that could go wrong, right?
So be careful of TikTok is what I tell people. And I'm not even kidding, because actually it's one of the most difficult things I have to deal with as part of my practice. Because people say, oh, this X, Y, and Z ingredient in this must be great, et cetera. And you perpetuate myths, right? And non-understanding of, actually what the regulatory landscape actually means.
Well, luckily I'm too old for TikTok.
Your kids. Thank you for the question. That was great.
Thank you for the nice talk. I have one question regarding the mechanism. Like you said that the CPDs are formed and the ProteXidine will protect from this thing, but how it will clear those, like in a damaged screen? So is there any?
So if we go back to here, let me go back to the slide. So we're talking about this is the protection part, right? Where you see making less, but then in terms of clearing the dimers, right? To clean this, so you are activating the nucleotide excision repair. So where's the? Okay, so this mechanism right here. The original, so back in the 90s, the thymine dimer by itself, if you dump it on the cells, you can activate nucleotide excision repair, right? So then when they developed ProteXidine here, right, again, stable molecule, and then they put it on and onto the cells that are damaged. Actually, you put it onto the cells, one, it can protect the cells from getting damaged, their own DNA, right? So it's a decoy in a sense there. But then once the ProteXidine gets damaged itself, it then activates that nucleotide excision repair. That's why, I'm sorry, not this slide, but that's why you see the clearance at the 24 hours. So it's a 1-2 punch. Protection at one layer, so the ProteXidine itself gets damaged. And damaged ProteXidine will cause the DNA repair enzymes to come through. And they showed that, with this stain for XPC, the enzyme over here. They have an increased expression of that repair enzyme in the presence of ProteXidine.
Has this been formulated into any sunscreens? And if so, have you seen the improved results with SPF testing?
Yeah, that's an excellent question, and I'll tell you something that was very shocking to me as a chemist, because I spend the last more than 35 years, me and my group formulating sunscreens, and I go way back formulating sunscreens. And like Dr. Issa was saying before, when we did the early testing on it, we tested ProteXidine in many different vehicles. 2 were sunscreens, so we had a regular sunscreen. Yes, this study here, we had a regular sunscreen and a tinted sunscreen. Then we had an anti-aging cream and another type of moisturizer. And I expected as a chemist that most of the CPDs would probably be prevented in the sunscreens. And I wanted to see the additional effect that ProteXidine has. And I was shocked when I looked at it and I realized, wow, the sunscreen itself doesn't have less CPDs than the moisturizer. So that was an epiphany moment where I stood there and I said, wait a minute, sunscreens are very, very, very good at shutting down the erythemal response. They're real good at stopping you from burning. They're not so good at stopping you from forming CPDs, which is really the real marker of cancer. or potential future skin cancers, I should say. So people put on an SPF 50, they think they're bulletproof. People think linearly, one-to-one relationship, so they think if 50 is about twice as strong as a 30. Neutrogena has a 70 out there. Don't ask me how they do that because the legal limit is 60. But in any case, what happens is, I should say I don't know how they do that, but in any case, what happens is people put the things on at these high SPF numbers and think they're bulletproof, and they stay in the sun. And they're not getting burned, but they're building up all the true markers of potential future skin cancer. So, and when you ask the industry, the government tracks money spent on public education. They're saying, well, we're educating more and more. And it's working. People are using more sunscreen than ever before. So why aren't the cancer rates dropping? And when they ask the industry says things like, well, it's the ozone layer, or people don't put it on the right way. And there's a lot of different excuses as to why things are not getting better. But you know, common sense would tell you a big chunk of why things are not getting better is because you shut off the erythemal response. People would normally go, oh my goodness, if somebody said, you're getting a little red, get out of the sun. That doesn't happen anymore. People stay on the boat, on the golf course, at the beach, whatever, and they're getting all that damage. As Dr. Issa mentioned before, they're building up those CPDs, and that's why, even though people are using more sunscreen than ever before, they're not protected. The other thing that he pointed out rightly and very importantly is that SPF only deals with UVB. And what happens now is there was a lot of talk about the so-called dark CPDs, and these are CPDs that form when the light source is removed. Well, you see from the slide here, 4 hours and 12 hours, that there's a development period to form the CPDs. They're ROS triggered, a big portion of them, reactive oxygen species. So what happens is most of them do form after the light source has been pulled away over the 4-hour period. So there's just a lot going on and a lot of misinformation out there, and I think ProteXidine is well suited to really protect the public and close that gap you see in treatment. Great question.
And just some of that, that formulation with sunscreen is actually in this paper as well, if you would like to read it.
And we're formulating a new sunscreen now, trying to make it a little bit more elegant and nicer feeling, but 2026.
That was a very good talk. My PhD was in DNA damage repair, so that was very exciting to see some biology from back in the day. My question, 2 questions. So if I come to you as a physician, do I just come to you and say I want that, or do I have to have an issue for you to?
You can come, so anybody can go to the dermatologist just to even discuss skincare, period. You know, they're like, I want to know what are the basics of what I should do to protect my skin, right? You don't necessarily have to have a problem in order to see a dermatologist. We welcome you anyway, right? So just to even get the basics of, you know, sun protection. Now, if let's say you've heard about this product, you can't right now go and buy it yourself online. It is dispensed at the dermatologist or at a doctor's office. And so the dermatologists have to know, you know, about this is why we're doing this and informing, so that they know to talk to you as the patient that there's this technology that can do pretty much everything you needed to protect yourself from the sun.
And what's the application rate?
The application like twice per day? Yeah. But sometimes I even tell people once a day is still better than nothing. You know, like people forget. We get lazy too. So. Well, on that note, I think if there's no other questions, thank you all for your attention and enjoy your day.
Thank you.
Thank you very much for sharing the story and the success. Those of you that don't know me, I'm Kevin Anderson. I work in the tech commercialization office, and I actually had the pleasure of negotiating and signing this license. And I figured I'd just give you just a quick flavor of the timing of this and how long an invention takes to actually get to the market. This technology was disclosed to our office in 2012. So it was June 6th of 2012. We filed a provisional patent on this. We haven't issued patent, obviously, on this technology. The first call was with Steve Hernandez, Abbas, and Bob and myself on, what was the date? November 2nd of 2018. So they came up to visit us on Valentine's Day of 2019. I think it was like 20 below. We were walking in the tunnels all around. We negotiated and executed the license on February 4th of 2020. So from February 4th, 2020 to 2024, 2025, the product, that development time. So 5 years, a little over 5 years to get to a product. So if you think about this, 12 years from the disclosure to a product is what this took. So quite a long journey. And throughout this, Bob and Abbas were awesome partners because we would go back and forth. And actually, this started with Rebecca. She was the first licensing officer on this technology. And as Bob said, she got promoted, so she passed it to me. And then I took it from there. So, but they had done a ton of work, and Abbas in particular, he went from knowing really nobody in the industry to now being seen as one of the key people in the industry. And it was through his efforts, really, of going to conferences, meeting people. We talked to everybody. Large manufacturing, chemical ingredients manufacturing companies, to all the big cosmetic companies, and even some pharmaceutical companies over the many phone calls and that. So it's really through a lot of the work that Abbas and Bob helping and supporting Abbas to do this that we were able to find topics. He found topics at a conference, and he would come to, he would come to the office, and he would had a spreadsheet of who he had talked to, what he had talked to them about, and he said, we got to talk to topics. And so subsequently we had that conversation, and here we are today with a number of products that are on the market and a successful commercialization. So I just want to thank you and Bob and Topics. This is exactly what we need in commercialization, is that partnership between the inventors, the researchers, and our commercial partners. So thank you very much for your commitment to this. With that, I think we're adjourned, done, right? Have a great day, everybody. Thank you.